Shailey Heller is our next speaker. Before coming to Scripps, Shailey was working in community engagement at Ocean Connectors, which is a nonprofit here in San Diego. She has a passion for community engagement and marine biology as well. And so her project also took us all the way to the other side of the globe to explore the role of people in science as well. Welcome, Shailey.
Day 29, -10 degrees Celsius. Light here is such a powerful element. The natural light is over the top beautiful and ever-changing. It's this month, October and April, that are Hilda's favorite months. She's outside filming right now a bit of this magic. We'll only have daylight for the next 7 hours, and every day this month, less and less, until October 26th. I think that's the date, when the sun has forever dipped below the horizon, below 5 degrees until February. It's a little bit hard to imagine, and even believe. I guess I should start taking my vitamin D, but I haven't yet cracked the bottle. I wonder how it will become in the dark. Poor Hilda, I write with a smile. This is an excerpt from a woman named Suniva Sorbi's journal on October 11, 2019. Driven by her love of adventure and citizen science, her and her expedition partner, Hilda Fallenstrom, became the first women to ever overwinter in a trapper's hut called Bumsaboo in Svalbard. Suniva's curiosity about how the darkness of the polar night would change her made me wonder how phytoplankton, photosynthetic microalgae, respond to the same conditions. To understand this question further for my project, I decided to analyze the gene expression of 10 phytoplankton samples collected by Suniva and Hilda during the polar night in Svalbard. Svalbard is an archipelago located at 78 degrees north near Norway. The location presents major challenges for data collection due to extreme weather and limited accessibility. But studying this region is essential. As the Arctic warms nearly 4 times faster than the global average, it's critical that we understand how its ecosystems will be impacted by these changes. This is where Hilda and Suniva saw the opportunity to make a major impact through citizen science. They decided to set up their own expedition to raise awareness about climate change and connect people all over the world to the vulnerable polar region of Svalbard. By reaching out to scientists, they planned a citizen science expedition that contributed to multiple research projects through collecting rare aurora, microplastic, ice core, cloud, polar bear, and phytoplankton data. My project focuses on the phytoplankton samples collected in partnership with Allison Cusick and Fjord Phyto. But why study phytoplankton? These microscopic organisms are photosynthetic and integral to understanding ocean health. They form the foundation of the marine food web, and their presence directly impacts the abundance of all other marine organisms in the ocean, including the seafood we eat. Additionally, they produce over half the oxygen on Earth through photosynthesis. Since phytoplankton rely on sunlight for photosynthesis, it was long assumed that they either died or entered a dormant state during the polar night. This assumption left a major gap in our understanding of how these 3 months of darkness affect seasonal phytoplankton blooms and ecosystem dynamics, a knowledge gap that further sparked my interest into this data-limited area of research. To understand how phytoplankton respond to the polar night, I first needed to understand the environment itself. 3 months of no sunlight, sub-zero temperatures, and the occasional Arctic storm. While the storm gives you a visual sense of the extremes, the data can show us how these variables change over time. On this graph, the x-axis represents time from the beginning of September to the end of December, and the y-axis represents the environmental variables of salinity, sea ice thickness, sunlight duration, and sea surface temperature, each in different corresponding units. The black dots are each one of the 10 sampling dates, and the red line marks the start of the polar night on October 28th. We see increases in salinity and sea ice thickness after the beginning of the polar night. This is because when sea ice forms, salt is expelled into the water, raising the salinity of the near surface water. Alternatively, we see declines in sunlight duration and sea surface temperature before the polar night begins, and then constant values at 0 hours of daylight and -2 degrees Celsius for sea surface temperature as the polar night begins. Now that we've seen the environmental extremes phytoplankton face, let's explore how they respond. Unlike animals, we can't observe their behavior directly, so we look inside their cells. Each cell contains DNA with genes that turn on or off depending on the environmental conditions. When active, a gene is transcribed into messenger ribonucleic acid, or mRNA, giving us a snapshot of the cell's current functions, a process called gene expression. By analyzing mRNA, we can identify which processes are active during the polar night. To measure this, I use transcripts per million, or TPMs, which quantifies how much mRNA is produced from each gene. But mRNA expression alone doesn't actually tell us what the gene does. That's where gene ontology, or GO terms, come in. GO terms provide a standardized way to describe gene functions across organisms grouped into 3 categories. First, biological processes, or the activities that the molecule helps the cell carry out. Next, cellular components, the part of the cell where the molecule is located. And last, molecular functions, the specific role done by the protein or RNA molecule. For this project, I explored all 3 categories, but for this presentation, I'll only focus on biological processes that are directly related to the environmental variables of salinity, sea ice thickness, sunlight duration, and sea surface temperature. I'll do this using a heat map. On this heat map, the x-axis represents the 10 sampling dates, and the y-axis represents the biological processes of response to cold, response to light stimulus, and response to salt stress. They all show clear responses during the transition into the polar night, marked by the red line. This format lets us see how gene expression changes over time, especially major shifts like the polar night. How we read the map is by the shades of the colors. So the darker blue means the higher expression, or the gene is more active, versus lighter blue is lower expression, and white is little to no expression. This part of my work focused on identifying patterns in how phytoplankton regulate their genes over time in response to environmental change. Let's zoom in on our first sampling date of September 24th. Genes related to light response and salt stress are highly expressed or upregulated in darker blue, while cold response genes are less expressed or downregulated in light blue. This fits with the environmental data because there's still sunlight and temperatures remain above freezing. After that strong initial response, gene expression in these stress-related processes steadily declines across the next 5 sampling dates, from October 1st to October 25th. The sustained decrease suggests that phytoplankton may be adjusting to new conditions, slowing cellular responses, and perhaps to conserve energy as light and temperatures drop. Just after the polar night begins, we observe a clear spike in gene expression. This likely represents final adjustments as phytoplankton transition into complete darkness, fine-tuning stress responses, and preparing for long-term survival in low-energy, high-stress conditions. It's important to note that even without any light, the cells are still responsive. In the last 3 samples, we're well into the polar night. At this stage, overall gene expression drops. On the heat map, this appears as the lighter colors are white, indicating that only minimal essential processes are active. This likely reflects metabolic downregulation as phytoplankton reduce energy use to survive sub-zero temperatures and total darkness. Field data like this is especially valuable for understanding how phytoplankton respond to extreme conditions and for shaping new hypotheses. Despite limitations and biases, my analysis produced preliminary results that open the door for future research into correlation versus causation. While most studies focus on spring and summer blooms, this project offers early insight into phytoplankton activity during winter. It lays the groundwork for understanding seasonal transitions and ecosystem dynamics. It also raises some new questions like, what genes are being prioritized? How does this influence spring recovery and bloom dynamics? What are the effects on the Arctic food web? Although we don't yet fully understand the mechanisms behind Arctic phytoplankton's winter survival, or how they may shift with changing ice cover light and nutrient cycles, we do know that even during the polar night's darkness, phytoplankton show signs of gene expression, demonstrating their resilience to survive. Just like Sunniva and Hilda's resilience during the polar night and commitment to citizen science made this analysis possible. However, their impact went far beyond gathering data. They also connected thousands of students all over the world to the rapidly changing Arctic environment by live streaming parts of their expedition. Their work is a reminder that science isn't just about numbers and measurements, it's also about people. Researchers and citizen scientists together represent the human side of science, each playing a vital role in advancing our understanding of the natural world. During her time overwintering in the Arctic, Sunniva said, "I believe so strongly in the two reasons we're up here, for science and connecting people. There needs to be deeper understanding around what in the world is going on out there, not just in the polar regions, but everywhere. And it makes us feel like we're doing something meaningful, even though we're out in the middle of nowhere, disconnected but connected." Whether it's a person overwintering in the high Arctic or a single-cell phytoplankton beneath the ice, both are responding to extreme conditions, and science is the tool that can eliminate these responses, revealing how life continues, even in the harshest environments. And I would like to thank my capstone committee for all of the help in this project. It would not have been possible without them, and also a big thank you to Sunniva and Hilda for collecting the samples, or else I wouldn't have had this data set. And there are a lot of people that I would like to thank. All might not be mentioned up here, and some are in the audience. But yeah, it's been a great year.
Great presentation, Shailey. So you mentioned that you also did heat maps for cellular components and molecular functions. Did you find anything interesting in those heat maps?
A great question. So I was new to this analysis before entering the program. So something I didn't expect was when we're well into the polar night on that last sample date, December 28th, that the chloroplast is upregulated, and that's where photosynthesis takes place. But then after some research, it makes sense because they're preparing for like immediately when light returns again, that they can start photosynthesizing and producing energy. And for molecular functions, I just really wanted to show my heat maps. So there's still like the clear spike on November 4th. And then I think the last three samples was kind of expected because more of the molecular functions that are more energy intensive with like proteins, repairing damaged DNA, and lipids are downregulated. And then stress response molecular functions and regulating osmotic stress and like salinity osmotic stress and pH are upregulated. Yeah.